Hello, college biology. This is photosynthesis part 3, phase 1, the light reaction. Let's get right to it. So, the light reaction primarily is conducted by a series of proteins embedded in the thylakoid. So here, let me move this. The chloroplast is being illustrated on the outside. So this is the outside of the thylakoid. The inside of the thylakoid is here, so we've been calling this the thylakoid space. It is sometimes called the thylakoid lumen. I will accept either. Moving me over here. And so we can see that there's a lot of proteins going involved here. Well, first of all, this story kind of goes from left to right in this diagram. And we have light, which kickstarts this whole process. Light is striking this photosystem, which is a series of chlorophyll proteins, enzymes that isomerize. They change their shape and perform a function when they absorb radiant energy. And so what's happening over here is we are splitting water into oxygen and some protons. And when we do that, we also pull an electron away. So now we have the electron. That's the point. The oxygen is a waste product and it gets released, but the electron gets passed to another series of proteins. And these proteins essentially pull in a hydrogen. So this is essentially, it's not the exact same type of hydrogen pumps that were in respiration, but it's still a hydrogen pump, and they pull in a hydrogen into the interior, so from the stroma to the thylakoid space. That's the first thing that the electron does. The hydrogen, the reason why this is occurring is because elsewhere down the membrane is ATP synthase. So as these hydrogens get pumped in, we create a high concentration of hydrogen. Those will diffuse back out through ATP synthase, and when that does, the ATP synthase isomerizes and it can create ATP. So that is sort of the first thing that is done with the electron. It pumps in hydrogen to create a concentration gradient so that there can be facilitated diffusion through ATP synthase to make ATP. Job 1 of the electron. Well, the electron's not quite done. It gets shuttled over here to this other photosystem, this other series of light reactive enzymes, and it uses the energy of light to kickstart some reactions that end up making NADPH, which is an electron carrier. So at the end of this, and that's it. That's what we do with the electron. It's done its 2 jobs. It's helped make ATP, and it has been put in an electron carrier. So that's it. That's the light reaction. So let's review. The chlorophyll photosystems in the thylakoid use light energy to take electrons from water, making oxygen. The electrons trigger the proton pumps, creating a high concentration of protons in the thylakoid space before the electron joins an electron carrier. Finally, the facilitated diffusion of protons through ATP synthase triggers ATP production. So at the end of the light reaction, we've made oxygen, which is a waste product that we get rid of. We've made ATP, which we're going to use in the next phase, and we've made electron carriers, which we're going to use in the next phase. That's it for the light reaction. I hope this helps. Struggle well.